Dear students, Assalamu alaikum. Today we are going to talk about our course Molecular Biology, course code BCHM 221. This is lecture number 15 of the series of our online lectures. The topic of today is post-translational modifications. This lecture is the continuation of the previous lecture because we talked about the overview of the process of post-translational modifications. Now we talk in more detail about the different processes. The outline of today's lecture is we will talk about the process of phosphorylation, then we will talk about the process of glycosylation. This will be followed by understanding hydroxylation, and then we will talk about ubiquitination. Then we will talk about S-nitrosylation. We will talk about alkylation and methylation, and acetylation, lipidation. So this then identification of PTMs, post-translational modification, how the post-translational modifications can be identified, the techniques. This will be the final point of our lecture. So now we move towards our lecture, phosphorylation. Reversible protein phosphorylation, principally on serine, threonine, or tyrosine residues, is one of the most important and well-studied post-translational modifications. Phosphorylation plays critical roles in the regulation of many cellular processes, including cell cycle growth, apoptosis, and signal transduction pathways. Phosphorylation is the most common mechanism of regulating protein function and transmitting signals throughout the cell. While phosphorylation has been observed in bacterial proteins, it is considerably more pervasive in eukaryotic cells. It is estimated that 1/3 of the proteins in the human proteome are substrates for phosphorylation at some point. Indeed, phosphoproteomics has been established as a branch of proteomics that focuses solely on the identification and characterization of phosphorylated proteins. Addition of phosphate group to a protein, principally on serine, threonine, or tyrosine residues, also known as phosphorregulation, plays critical role in cell cycle growth, apoptosis, and signal transduction pathways. Here you can see non-phosphorylated protein. This is the OH. ATP enter either on serine threonine with the help of this enzyme, serine threonine kinase. It comes out as ADP, and then you can see the protein gets phosphorylated. This triphosphate converts to diphosphate. This one phosphate is stated here. Now the protein is phosphorylated. This is a reversible process, and it can, with the help of the same enzyme, serine phosphatase, with the help of another enzyme, this is serine threonine kinase, and with the help of another enzyme, serine threonine phosphatase, when water enters, inorganic phosphate can go out, and it can again convert it into non-phosphorylated protein. So this process of phosphorylation can convert non-phosphorylated protein to phosphorylated protein, and serine threonine phosphatase can remove the phosphate and it can convert it again into a non-phosphorylated protein. Phosphorylation of amino acid residues is carried out by a class of enzymes known as kinases that most commonly modified side chains of amino acids containing a hydroxyl group. You see here, This is the hydroxyl group. This is serine threonine kinase. It phosphorylates the protein. So phosphorylation of amino acid residues is carried out by a class of enzymes known as kinases that most commonly modify side chains of amino acids containing a hydroxyl group. Phosphorylation requires the presence of a phosphate donor molecule such as ATP, GTP or other phosphorylated substrate. In this situation, you see that ATP work as the donor of phosphate. It can be GTP or other phosphorylated substrates. Serine is the most commonly phosphorylated residue, followed by threonine and tyrosine, because they have their OH group that can be phosphorylated. Removal of phosphate group is carried out by the phosphatase enzyme, and thus this forms one of the most important mechanism for regulation of protein. Here you can see that serine 3 phosphatase, it removes the phosphate and it is again converted into non-phosphorylated protein. So it can be either phosphorylated or dephosphorylated. Glycosylation, another process of post-translational modifications, is glycosylation. Glycosylation involves the enzymatic addition of saccharide molecules to amino acid side chains. This can be of two types: N-linked glycosylation, which links sugar residues to the amide group of asparagine, and O-linked glycosylation, which links the sugar moieties to the hydroxyl groups of serine or threonine. Suitable glycosyl transferase enzymes catalyze these reactions. Sugar residues that are attached most commonly include galactose, mannose, glucose, N-acetyl glucosamine, N-acetyl galactosamine, as well as fucose. Now, O-glycosylation adds serine residues. Here is the serine residue. UDP and glucose enter, UDP goes out, glucose attaches. An example of how the process of glycosylation takes place. The covalent attachment of oligosaccharides, addition of glycosyl group or carbohydrate group to a protein, principally on asparagine, hydroxylysine, serine or threonine. Significant effect on protein folding, conformation, distribution, stability and activity. It is natural because the attachment of any of these groups, oligosaccharide, carbohydrate, or glycosyl group, it brings a structural change which will affect the overall structure of the protein, and it can affect the protein folding, it can affect the conformation, distribution, stability and activity, because protein structure and function are very much related to each other. Hydroxylation. During the formation of collagen, the amino acids proline and lysine are respectively converted to hydroxyproline and hydroxylysine. This hydroxylation occurs in the endoplasmic reticulum and requires vitamin C. Ubiquitination. Ubiquitin is a small regulatory protein that can be attached to the proteins and label them for destruction. Effects in cell cycle regulation, control of proliferation and differentiation, programmed cell death or apoptosis, can function in DNA repair, immune and inflammatory processes, and organelle biogenesis. You see here the ubiquitin the help of ATP, AMP plus PPI goes out. The butene attaches here in another cycle. E2SH and E1SH goes out. E2 attaches here. Now in the next step, this is the substrate. The ubiquitin takes here, and in the next step, you see this leakage is formed, and this is the substrate, and this is the ubiquitin attachment. This overall this diagram is showing the ubiquitin cycle. Now another process of post-translational modifications is the S-nitrosilation. Nitrosyl NO group is added to the protein. NO, a chemical messenger that reacts with free cysteine residues to form S-nitrothiols used by cells to stabilize proteins, regulate gene expression. See the process of nitrosylation, C2SP. This is the NO attaches here, and in another way, this with this O2 radical can form this OONO, and it can attach here with tyrosine nitration. This is metal nitrosylation, and this is S-nitrosylation. So it can perform these different types of this NO in different types of nitrosylation. This is tyrosine nitration, this is metal nitrosylation, and this is nitrosylation. 3 different types. Then we have alkylation or methylation, addition of methyl group to a protein, usually at lysine or arginine residues, binds on nitrogen and oxygen of proteins. Methyl donor is S-adenosylmethionine, SAM. Enzyme for this is methyl transferase. Methylation of lysine residues in histones in DNA is important regulator of chromatin structure. Here as S-adenosylmethionine is converted into S-adenosyl homocysteine in alkylation at lysine residues. The lysine residues, this is S-adenosylmethionine, and then it goes out as S-adenosyl homocysteine, and this is how this product is formed. See the methyl group attaches here, resulting in the process of alkylation or methylation. Then another process of post-translational modifications is the N-acetylation. Addition of acetyl group to the nitrogen. Histones are acetylated on lysine residues, and the N-terminal tail is a part of gene regulation involved in regulation of transcription factors, effector proteins, molecular chaperones, and cytoskeletal proteins. Methionine aminopeptidase (MET) is an enzyme responsible for N-terminal acetylation. This is N-acetylation at lysine residue. This is the lysine residue. COASR enters and COASH goes out, resulting in N-acetylation at lysine residue. Now, another process of post-translational modifications is the process of lipidation. Lipidation, attachment of a lipid group such as a fatty acid covalently to a protein. Now, you might have heard about lipoproteins. Lipoproteins are the ones in which lipids are attached to the proteins. So these lipoproteins are formed as a result of this process of lipidation. Lipidation attachment of a lipid group such as a fatty acid covalently to a protein. Normally, in the process of translation, we have only polypeptide sequence that consists of only amino acids, but in case lipoproteins are formed, lipidation is done. This is a form of post-translational modifications. In general, lipidation helps in cellular localization and targeting signals, membrane tethering, and is a mediator of protein-protein interactions. So, with the help of this process of lipidation, when the lipoproteins are formed, they can perform these various functions, including targeting signals, membrane tethering, and mediator of protein-protein interactions. Now, the process of post-translational modifications can be monitored by various methods. One of the methods is by mass spectrometry. Mass spectrometry tells us about the mass of protein. So if we want to know about any post-translational modification that has happened to a polypeptide, we can use a mass spectrometry, and there it will tell us about the change in the mass of the polypeptide, and through this way we can get to know about what type of structure is attached with the polypeptide, and this is a way to confirm or to tell us about how much or what type of molecule or species has got attachment with the polypeptide. Then another way of identifying post-translational modification is by utilizing high performance liquid chromatography analysis. High performance liquid chromatography analysis is helpful in identifying about post-translational modification that has happened to a protein, a newly formed polypeptide, or incorporation of radioactive groups by addition to growing cells. Radioactive groups can be incorporated to growing cells, for example 75 EC labeling and chromatographic isolation of proteins. They can give us idea about post-translational modifications. Then we have another type, it's called antibody cross reactivity. Antibody cross reactivity can also be helpful in identification of post-translational modification. For example, antibody against phosphotyrosine is an example of identification of PTMs or post-translational modifications. Then there is another way called polyacrylamide gel electrophoresis, or PAGE. Polyacrylamide gel electrophoresis tells us about the molecular mass of a protein. So with the help of polyacrylamide gel electrophoresis, we can get an idea about what type of post-translational modification has occurred. So this is the summary of the different techniques which can tell us about the type of post-translational modification that protein has gone through. So I hope you got an idea of the different types of post-translational modification that a protein has to pass before it is converted into a proper functional protein. Thank you very much for watching, and if you have any questions, please feel free to contact me. I will be happy to answer. Thank you again.